Alright, so I'm not going to start you off with an essential question yet, just because I want to go into some really cool mechanisms for evolution. So just some really cool and interesting things that are more, I don't know, they've developed more in the last several years since I've been teaching this, so I want to share them with you. But here's what we've got. So genes that control development play a major role in evolution. These are called homeotic genes. And these are a really cool set to know, because these are the master control genes. So they determine basic features such as where pairs of legs and wings and things develop on a fruit fly. So profound alterations in body form can result from changes in homeotic genes or how and where they're expressed. So homeotic genes I always think of are kind of like blueprints. So they're kind of like your blueprint genes. They say that the arm is supposed to go here, the leg is supposed to go here. They really lay out the blueprint of where all the correct body parts are supposed to go. And when you mess with that, you mess with certain things. Like here's a homeotic gene that says that this particular ventral fin is supposed to be here, but in this particular one it's absent. So altering that homeotic gene, that blueprint gene, gives us a removal of that kind of ventral belly fin. So, interesting things. Alright, so most complex structures have evolved in increments from simpler versions having the same basic function. So it's kind of like a process of refinement. And there was one class period that said that things get more complex. And as a general rule of thumb, most structures do seem to get more complex over time. But that's not always the case, which I think I mentioned in whatever class period said that. But sometimes we can trace the origin of evolutionary novelties to the gradual adaptation of existing structures to new functions. So you can take stuff that we already had and give them new functions. Okay, so that's sort of new things, old things, new functions. I'll sort of write that in that manner. As an example of the process of gradual refinement, considering the whole kind of camera-like eye situation that we see in vertebrates and squids, which are invertebrates. It's fascinating. So although eyes of vertebrates evolved independently from squids, both evolved from a simple ancestral patch of photoreceptor cells, and through a series of incremental modifications have benefited their own at each stage, or their owners at each stage. So, there appears to have been a single evolutionary origin of light-sensitive cells. So that would be kind of photoreceptors, because photo is light. So that's where we get the photo, and then sensitive is kind of the receptors or the receivers. So our photoreceptor cells kind of have a single origin with all these animals, but they share the same master genes that regulate eye development. So we can see sort of what happens here, and this is, you don't have to write all of that, but it's just fascinating stuff. Here's kind of the original photoreceptors, like these particular cells could kind of receive and understand light. But then over time, you end up with an eye cup that you would see in abalone, and then it becomes a little bit more fluid-filled with a pinhole that we would see in nautilus and a marine snail. Now we're starting to get a very primitive lens. And then by the time that we get to something like a squid, it's a very complex lens. And actually, this is a very similar kind of eye structure that we would see in mammals. See the whole lens and the whole optic nerves and the retinas, all that's there. Alright, so evolutionary novelty can arise when structures that originally played one role gradually acquire a different one. So when we change the roles, that's where we get some new things. So such structures that evolved in one context become co-opted for another function are called exaptations. You don't have to write that one down, but it's just a cool thing to know. So feathers that first function for insulation and were later co-opted for flight. And then flippers of penguins that first functioned for flight are now co-opted for underwater swimming. So an exaptation is just acquiring another function. And I really think penguins is a good one, because they're not really into the whole flying business. They're more for swimming, and so those kinds of fins have been kind of exapted to be for swimming. something you can think about. The fossil record also shows the trend of many in evolution in many species, for example, towards a larger or smaller body size. So sometimes smaller can be more complex. But the evolution of horses reveals a potential misunderstanding. If we select only certain species, it appears that there would be a general trend towards a reduction in the number of toes to larger size and teeth for grazing. However, we consider all the known members of this family tree, it kind of vanishes. Again, just some other things to kind of look at. So over time, millions of years ago to present, you see it sort of gets larger, but then not necessarily, because then we've got sort of a weird situation happening in here. So we don't necessarily have all the right information. So you need to include everything. There's a lot over here and maybe a lot over here that would also have to be present for us to kind of figure that out. And honestly, so looking at all this stuff and looking at it over time is how we build kind of these trees. So if you've seen the trees of relationships, that's what we're going to do. We take all these similar things and we're going to put them together to make a tree. That's coming up. So branching evolution can lead towards real evolutionary trends. The species selection model for long-term trends compares species to individuals. So speciation is their birth, extinction, their death. So speciation, birth, extinction, death. And the new species that diverge are kind of like their offspring. So unequal survival of species and unequal generation of new species plays a role in macroevolution, similar to the role of unequal reproduction and microevolution. Lots of things. Again, it's like, is it fitting in? Is it the best fit for that environment? So evolutionary trends can also result directly from natural selection. For example, when the horse ancestors invaded the grassland that spread into the mid-Cenozoic, there was also a strong selection for grazers that could escape predators by running faster. So if you're fast, but you also like to eat grass, you're doing pretty good. So whatever its case, it's important to recognize that an evolutionary trend does not imply that evolution progresses towards a particular goal. Evolution is the result of interactions between organisms and their current environment. So somebody asked about that as, can we ever fashion a perfect organism? No. And that's because that environment is always changing. I mean, heck, I think look outside. That's a perfect example of our environment sort of changing, whether that's natural or not natural. The importance is that it's changing. And so that's a changing environment that different insects, different animals, whatever it is out there, they all have to adapt to it. So evolution is the result of interactions between organisms and the current environment. That's super key. That's why we're not making ideal ones or perfect ones or whatever. It's because it has to adapt and fit with whatever environment is there. So it's super key. All right, we're at about 8 minutes, and it would be a great time to take a break if you needed to take one. And then we'll move on to kind of phylogeny and the tree of life. So we're talking about all the naming schemes and taxonomy. All right, so this one has an essential question that we should really look at. And it's, what's phylogeny in the tree of life? There you go. What's phylogeny in the tree of life? But we should also kind of include taxonomy. And taxonomy, because that's going to be something that we're going to talk about. That is a really big one. Taxonomy. So there you go. So it's what's phylogeny in the tree of life, and then taxonomy, because that's kind of all we're going to cover in here. There's quite a bit to it, but it's, I think, stuff you've seen before. We're just going to elevate it a little bit and give it more terms. So phylogeny is the evolutionary history of a species or a group of species. So it's going to sort of give us our sort of family trees. We'll say, like, it's going to be like family tree of species. Okay, so phylogeny can be inferred from the fossil record, morphological homologies. Again, it looks like a whole lot, but it's just morphology is like the more for the form, or that's form, or kind of looks like. What does it look like? And homologies are still similarities. So break that one down. Looks like a lot, but it's just similarities in form or similarities in what they look like. Same with homologies, or again, homo is same, so we're talking similarities in molecules. So that's a lot of words, but it's not that hard. Phylogeny can be inferred from the fossil record, similarities in what they look like, and similarities in what their DNA or molecules are like. Okay, so there you go. Homologies are similarities. So just a little, if you needed an extra on that one, homologies are similarities due to shared ancestry. That's key on that one. So evolving from the same structure and a common ancestor. Okay, so homologies we think of, oftentimes what it ends up being is the same structure. Oh my goodness. Different function. Okay, and so when you know they're the same structure, you know they come from the same ancestry. This is going to be important. So generally, organisms that share similar morphologies are more closely related. However, some similarities are due to similar adaptations in a common environment. We call that convergent evolution because it kind of converges or comes together. And so a similarity due to convergent evolution is an analogy because they're going to have different structures, but same or similar function. See how these two things are a little different? Is that homologies have sort of the same ancestral structure, but it can have a different function, whereas with analogies, they'll have completely different structures, but the same function. If you're like, what are you talking about? Like, oh, this is probably a bad example, but these two guys aren't actually related. Because they're both diggers and live underground, they have similar functions in feedage. I want to say it's a, God, I don't remember. This one's definitely a mole, and this is sort of similar to a mole, but they both have short hands for digging. So this is what, this would be an analogy, because they don't actually have a common ancestor. But they do have a similar environment of digging kind of under soil. There's a better one. Let's give you a better one. So analogies can also be like, I'm going to draw you some simple structures so you can see these two, because you may not be familiar with the mole and the, I think it's like a version of a rat, but I can't remember. Anywho, you can also think like, there you go, there's a little dragonfly, or a butterfly, doesn't matter. But then also you can have like, see if I can draw this properly. Oh well, you know what I'm getting at. It's a bat. So dragonflies and bats both are adapting to the environment of air, and so they both have wings, but bats have bones in their wings, and then dragonflies just have these kind of thin filamentous sort of wings. So two similar structures, but different, different structures, but the same function for flapping. There we go. I'm getting the words straight today. All right, systematics. This is the stuff that has to do with taxonomy, which you probably know, but don't always think that you do know. So systematics is a discipline of biology that focuses on classifying organisms and determining their evolutionary relationships. People who study that are systematists. It's cool because they get to deal with a variety of things if that's what you're into, but I have yet to meet a systematist that wasn't a straight up weirdo, and that's fine. Everybody's gonna have their place. I've met a few. So, classifying organisms and determining their evolutionary relationships. One of the earliest people that we know of for some of this stuff his name was Carolus Linnaeus. So Linnaeus is the dude who originally came up with taxonomy. And this is a system of naming and classifying species. So both of these are definitions that you sort of need to know. All right. So biologists assign, so this is part of actually taxonomy, This is the whole naming scheme that Linnaeus came up with. So this is the taxonomy. So we assign each species a two-part name, or binomial. Binomial meaning name. So it has the genus, and then it has a unique part at the end of it, sort of like a last name. Okay, for each species, that's sort of a unique part. So it's got its genus, and genus are more common. Like, you know, my first name is Kelly. That's pretty common. That's like there's lots of people that have Kelly as a first name. But the unique part, the specific epithet, is my last name. So Royce. So Kelly Royce, that's my name, right? So the Royce tells me specifically who that is. But my name overall is my full name. So the species name actually includes both. So if I were a species, Kelly Royce, that's my species name, is both names. Not just Royce, not just Kelly, but both. Okay, so genera are grouped into progressively larger categories, and each is called a taxon. So we'll look at it. So we'll look at this. Just look at that. That's a face right there. That's the kind of cat that you want to use in this example, because I don't. He is upset that he is here today, and that is okay. But here's the thing, and we learned about these guys way off in the kind of early of the last semester, is that we have 3 domains: bacteria, eukarya, and archaea. So it goes from here. And I actually don't like, usually I go down with them, but that's okay. We'll go up from here. So the biggest is the kingdom, and so this is kingdom Animalia, and then under that we have phylum, or phyla, the chordata. Then we have class, Mammalia, order, Carnivora, family, Felidae, and then genus. Notice genus is the first name, Felis, and then the species is both names. So Felis catus. So catus is the specific epithet that we just learned about. So specific epithet is the catus part. But both Felis catus make up the species name. So one thing that you will need to know is you'll need to know the kingdom, phylum, class, order, family, genus, species, business. I just know them from having done it long enough, but I've heard King Philip came over for good spaghetti, I think, or grape soda, or whatever. King Philip came over for great spaghetti. I don't know, whatever. Come up with your own weird thing. The weirder it is, I don't know why, but the weirder it is, the better it sticks. So come up with something. All right, so biologists traditionally use phylogenetic trees. So that's kind of the end of the naming scheme. We're going to practice that thing. We'll use dichotomous keys and stuff like that, which you don't know that you know how to do, but you do. So we're going to practice with those. But here's where we're at. So we typically will use phylogenetic trees to depict hypotheses about the evolutionary history of species. So these are branching diagrams that you've seen before that reflect a hierarchical classification of groups nested within more inclusive groups. So it's sort of like this is the species kind of family tree business. Okay, so the phylogenetic tree is going to be sort of like species family trees. It's like their own version of Ancestry.com. They're figuring out where the little leaves are and where their ancestors go. So that's basically what it is. So phylogenetic trees indicate the probable evolutionary relationships among groups and the pattern of descent from the last common ancestors. Okay, let's see if I can move this thing forward. There we go. So there's a couple of ways that these are represented, but overall, we could look at it this way. So Carnivora, or Carnivora, is our order, and then we've got families kind of branching off of these. So apparently Felidae kind of went its own way, but we have these other two. Right? And if we looked at Mastelidae, it would branch into the genus of Mastella and Lutra, and so that would give us a weasel and an otter. And so we can sort of see that the actual common ancestor of like the otter and the cat, if we sort of follow those lines back, it's way back to Carnivora before these guys have a common ancestor. So that's sort of what we're looking at here. For these guys, like if we follow let's say like a wolf, and we followed that back to the otter, go back here, where those guys actually meet would be at this node. So we'd assume that there's sort of a common ancestor right here, that these guys branched off from. If you look at like the coyote and the wolf, well, their common ancestor is right here. So it's clearly more recent than some of these others. So they're more closely related. And we know that too because they both share the genus of Canis. And so they're both similar in the same genus. They're just different species. So they're kind of related. They're like cousins. If that sort of makes sense, drawn all over that. Cladistics is the most widely used method of systematics. It groups organisms into clades, and each clade should be one monophyletic group. So one sort of evolutionary whatever. So like we could think almost sort of like one family. We want them to be one family. And we want it to be including all ancestral species and all of its descendants. So sort of like a family tree. Think about like your nuclear family. A monophyletic group would sort of be like you and your parents and your grandparents. But if we're including your grandparents, we're also including like your aunts and your uncles. We're also including your cousins. Like you have to include, like if we're taking your grandma for a monophyletic group, we have to include all of her children, all of those children's children, all of those things have to sort of be included to call that a monophyletic group. It's like that one family group. In the same way with species, if we're going to include, you know, carnivores, we've got to, so if we look back, if we're going to include carnivores, we have to include all these, but if they're missing some cousins in between, we need all of them to come together too. If we're just saying maybe the Canis one, well then we just need to include all the things that fit under Canis, all their little cousins. We don't need to include them, like the cat and the otter and stuff. We just need to include those. So a monophyletic group includes the ancestral species and all of those descendants. Where do you want to start though? That makes sense. So cladistics is based on a Darwinian concept that organisms share characteristics with their ancestors and differ from them just a little bit. So there's two main characters. There's shared ancestral characteristics and shared derived characteristics. So ancestral ones we'll see that are kind of ones that originated in an ancestor, and shared derived are unique kind of novelties. Again, we'll build a little tree, and so we'll sort of make more sense as we go along and actually use some of these terms. So you might jot down in-group and out-group, but this won't make sense until we build a tree, and we'll build one together. It's a tough thing, but we're going to build it. So an important step in cladistics is the comparison of the in-group, the taxa whose phylogeny is being investigated. So like if if it's grandma, and the out-group would be kind of who diverged from the lineage leading to the members of the in-group. That one's kind of a weird one to talk about. Again, this should make more sense. You can always write these definitions down, just a quick kind of version of them, but it's going to make more sense when we build the phylogenetic tree. Okay, so as an example, we consider a frog representing the outgroup. So we'll have a frog as the outgroup, and then we'll have four other tetrapods, or four-feeted things, that will be the in-group. So if we have a presence of a characteristic or absence of a characteristic, so 1 would be present, 0 is absent. Here's sort of what that looked like. Here's our frog, and all these things are going to be absent. But if we take the iguana, it has an amniotic egg, that's present, but not hair, or it doesn't hold babies very long or have a long gestation, like a long pregnancy. Duck-billed platypus, it's got amnion and it's actually got hair and mammary glands, but doesn't really hold a baby and doesn't have a long pregnancy. Kangaroo, a little bit more. Beaver definitely has all of them. So you can actually build the tree this way based on figuring out ones and zeros, then you can line them up. So we kind of say, if we look at it both ways, the amnion, the frog doesn't have it, but everybody who follows does. So everybody after this tick mark is going to have it. Iguana, duck-billed platypus, kangaroo, and beaver. Just like we see here. Hair and mammary glands, we'll see that in the duck-billed platypus, the kangaroo, and the beaver. So that's sort of how we have to arrange things. Again, strange and looks like a lot, but we'll go through it together, and doing so will help us understand how to read them better. The only way to do it. All right. So in our example, the phylogenetic tree is constructed in a series of branch points that we call nodes. Again, we'll work through these with a new set of derived characters. So when constructing a phylogenetic tree, we use a thing called parsimony, which looks for the simplest explanation for observed phenomenon. So that's one that might be best to know. So parsimony is just like the simplest is probably the most correct answer. I don't know if you guys have lived long enough to know that, like, I don't know that everything works off parsimony, because I've had some real complicated stuff happen in my life. So I always wonder about parsimony. We base a lot of things off of parsimony, but life is complicated and wild, so I don't know. But it's a thing that we kind of go through to kind of make some observations and make some comparisons. So it works for our kind of like making comparisons and making sense. Does it mean that it's completely true all the time? No. But it helps us get a better idea, and that's all that we need to work with. So there's parsimony for you, which is a good one to know. All right, systematists use many kinds of evidence. However, even the best tree represents only a likely hypothesis. So there you go, based on evidence. So it's just a hypothesis. All right, it's just our best guess. All right, as new data accumulate, hypotheses are revised and new trees are drawn. Phylogenetic trees of reptiles show crocodilians are the closest living relatives to birds because they all have four-chambered hearts, so that's morphological. They sing to defend their territories, believe it or not, and they do build some nesting sites. So these traits likely present in the common ancestor of birds, crocodiles, and dinosaurs. Maybe that Archaeopteryx if you haven't looked at it. So this would be sort of where their common ancestor is. So we're looking at kind of crocodiles, pterosaurs, ornithischian, birds, that kind of thing. All right. So we can see we get fossil records. Again, fossils are helpful for us figuring this stuff out. But we get some ideas with front limbs, hind limbs, and the fact that there's eggs in there. Pretty gnarly. We also have molecular systematics, and it uses DNA and other molecules to infer relatedness. So we've sequenced more than 110 billion bases of DNA and thousands of species. So an enormous database has fueled the boom in the study of phylogeny, good lord, and clarified many evolutionary relationships. So the more recently two species have branched from a common ancestor, the more similar their DNA sequences should be. So the longer two species have been separated on evolutionary paths, the more their DNA is expected to be different. That should make sense. So molecular evidence has also begun to sort out relationships among the species of bears. So we could sort of look at it as the panda, weasels, raccoons kind of have an ancestor back here at this particular node. There we go. Mark that thing up here. The giant panda is kind of its own. Pandas I don't understand, but they're cute. But I don't understand them behaviorally. Back there. If we looked at a few things, the American and the Asian black bear have a common ancestor here. Polar bear and the brown bear. We know it makes that weird growler bear or whatever. It's in there. So you can see kind of how closely these things are related to each other. Just pretty cool, based on DNA evidence. So different genes evolve at different rates. DNA coding for ribosomal RNA changes pretty slowly. So it's useful for investigating these relationships. In contrast, mitochondrial DNA is kind of all over the place. It does rapidly. It's really used to investigate really recent events. So we know about how these things evolve and how they change over time, and that can let us know what kind of code we should be looking at. All right, here's a remarkable commonality of molecular biology. It demonstrates that all living organisms share many biochemical and developmental pathways. And it provides overwhelming support for Darwin's theory of descent with modification. So we just get a little extra check mark, like, good job, Darwin. One of the last things we'll look at is molecular clocks. So we rely on genes that have a reliable average rate of change. And we can actually sort of calibrate those in real time, by the way. You might want to just write that bit down. But you can actually calibrate them by graphing the number of nucleotide differences against the dates of evolutionary branch points that we actually know in the fossil records. So we can use those to estimate the dates of other evolutionary episodes that are not documented in the fossil record. So we can use genes to tell us how old things are. Just the more technology we have, the crazier this gets. All right. Again, molecular systematics and cladistics are kind of a remodeling. So we now have this 3-domain system that we didn't used to have. We used to start at kingdom. When I was in school at your age learning all this stuff, we started at kingdom. And now we push it back and we start at domain, and we say we have bacteria and archaea, and now eukaryotes forming their own called eukarya. Eukarya have then plantae, fungi, and mammalia. I think Protista is polyphyletic, so you can't really use that one at this point. But, anywho, molecular and cellular evidence indicates that 2 lineages of prokaryotes, bacteria and archaea, diverged very early in the evolutionary history of life. So archaea is more closely related to eukaryotes than bacteria. Just if you wanted to know that little tidbit. Stomach's growling. I don't know if you guys can hear that. I'm sorry. So the most recent ancestor of all living things would be back here, and the gene transfer between mitochondrial ancestors and the ancestors of eukaryotes would have happened somewhere in here. And then a gene transfer between chloroplasts to the ancestors of green plants. Isn't that fascinating? We can kind of look at the parts changing. I'm wrapping it up, I promise. All right, you can sort of see archaea and eukarya. If we put the tree of life together, it's absolutely massive. But people have been working on trying to do that for quite some time. So I know that went a little bit longer. As always, pay attention to the details about what bits you need to know and which ones you don't. If you have any questions at all as to what's a priority here, shoot me an email and we'll get it figured out.